Hello. So we're going to talk about acid-base physiology today. So we're kind of continuing from our renal chapter, and we're kind of gonna keep talking about renal influence, but we're also going to kind of mix that with the respiratory influence that we've also talked about, and then just pH in general. So we're going to kind of put a few things together and talk about how we maintain pH or blood gases, arterial blood gases, like our carbon dioxide levels, our oxygen levels, and then and that effect on pH. So that's kind of where we are now. So we have to talk about what pH is. So this is something we learned maybe in high school chemistry, maybe college chemistry, and maybe we don't remember it. But either way, we're going to go over it again quickly here. So an acid contributes hydrogen to a solution. So this is really all about hydrogen. So hydrogen, remember, hydrogen has one proton and one electron. So the positive ion would lose that electron and it would just leave a proton. So that's why we sometimes just refer to hydrogen ion, the positive hydrogen ion, as a proton, because that's all that's left, because it has lost its only electron, and that's what it's left is, as a proton. So we can call it hydrogen ion, we can call it a proton. And that becomes relevant because the drug that blocks protons or hydrogen acid in the stomach are called proton pump inhibitors. So that's why we need to know that's also called a proton. Okay, a base decreases hydrogen in a solution. Sometimes we say that's an electron or a hydrogen acceptor, and so more hydrogen means more acidic, less hydrogen means more basic or alkaline. Okay, so that's another word that we use for that. So the pH is the negative log of hydrogen. So of the hydrogen concentration, those little square brackets mean concentration. So the negative log of the hydrogen concentration. Now hydrogen isn't in the body at very, very high concentrations. It's 0.000000 something, okay, usually. So with logarithms are kind of the opposite of exponents. If you think about exponents, like something to the 10th power means, it's like 10 numbers, like it's like a billion or 10 million or something like that. So it's very high numbers when you talk about exponents. So if we go the other direction and we say the negative 7th, then we're talking about 0.0000, small numbers. So logarithms deal with small numbers, and when we use a logarithm to kind of give the value of the pH, that means that even though something is 0.000001, we can just say it's 7. Okay, and so that's kind of how we end up with this logarithm, or this negative logarithm, to give value to our hydrogen ion concentration. So it actually gives us a number of the concentration, but it's called pH, and it's a little easier to read. It kind of feels like it goes in the wrong direction, because a lower number is going to mean a higher concentration. So a higher concentration of like 0.00001 is actually more hydrogen than this one, which is 0.00001, but then your logarithmic value may be 1, 2, 3, 4, 5. So your pH here might be 5. Okay, which means that it has more hydrogen and a lower number, which means it's more acidic. Okay, and we'll see a chart of that in a couple of slides from here. So we also are going to talk about buffers. Buffers minimize the changes of pH. So when you add acid to a solution, it tends to make it more acidic. When you add bases, it tends to make it more basic or more alkaline. And so buffers will kind of accept those hydrogens when you dump them in there, and they will accept or donate a hydrogen to a base when you put it in there. So it kind of buffers it, obviously it's in the name. And that's kind of what we expect to happen with a buffer. So let's see, we're still talking about hydrogen in general. So hydrogen may be formed from a reaction with carbon dioxide and water. So we have seen this. So the carbonic anhydrase enzyme will cause carbon dioxide and water to kind of come together and form carbonic acid. Carbonic anhydrase, by the way, just a little trivia for you there. It is the most efficient enzyme ever found. So that means that it enzymatically causes a reaction to happen, like, I can't remember, but it's like 1,000,000 times faster. Okay, I might be exaggerating or I might not be. I'm not sure because I honestly can't remember, but it's very, very fast. And so even though carbon dioxide and water may do this naturally at some point, without carbonic anhydrase, it'll take them probably years. Okay, so it's a very important enzyme, this carbonic anhydrase. But anyway, so carbon dioxide, water will mix together to make carbonic acid, and then the carbonic acid can dissociate into that hydrogen, which is going to cause that pH to get lower, the concentration to get higher. Okay, so we've seen this equation, we know it, we love it. Okay, so hydrogen is also produced from protein and phospholipid catabolism. Lots of things can cause hydrogen to be kind of excreted out. So we're going to kind of run into some others of those. In fact, we could kind of talk about them here. So hydrogen and ion and pH balance in the body is what we're kind of trying to maintain. So if we look at this top bar here, we're seeing that we have fatty acids, amino acids, okay, so those all have hydrogens that can kind of fall off and increase the hydrogen ion concentration. So fatty acids, amino acids in our diet can start to kind of drop our pH or increase that hydrogen. We saw our reaction with carbon dioxide and water, and then to cause hydrogen. And then lactic acid. Lactic acid is caused from, well, one of the things is aerobic respiration. Okay, can cause a buildup of lactic acid. We see that, or we may have heard of that, like with you, if you work out, do anaerobic exercise, sprinting, things like that, that you may have a buildup of lactic acid. Well, it also happens in things like shock. It's just because there isn't enough oxygen. Okay, so if you have a hypoxic situation somewhere in the body, or if you have just very, very low blood pressure, such as what may happen with shock, then you may have again a buildup of lactic acid. Keto acids from burning fat, so you might see that with DKA or diabetic ketoacidosis, where the cells can't access sugar because insulin isn't taking the sugar into the cells for it to use. And so it's an interesting disease. So the sugar in the blood is high, but it can't get into the cells. And so the cells have to start burning fat. And when they burn fat for all of their energy, then they start producing things like beta hydroxyl, acetone, and the acids that are associated with the fatty acids, those ketones. Okay, and there are a number of different ketones. Okay, so ultimately what happens is that we have these things that are trying to, in this case it's all hydrogen, that are trying to really drop our pH to be lower, okay, so make things more acidic. Our normal pH is, well, this says 738 to 7.42. The number that we all need to know is 7.35 to 7.45. Okay, so it gives us that kind of theme of 3.5 to 4.5. We see that with sodium, 135 to 145. We see it with carbon dioxide, 35 to 45, and our pH, 7.35 to 7.45. Okay. All right. Just in case you weren't aware of that, I don't know. It's a trick that helps me. So we have a lot of hydrogen coming in here, and we need to do something about it. We have the immediate line of defense, our buffers. So bicarbonate is kind of sitting in our blood, and it acts as a proton acceptor or a hydrogen acceptor. So if you have enough bicarbonate, then that will accept those free hydrogens, and it'll kind of neutralize it. Okay, so that's found in extracellular fluid. We see, and also there are phosphates in extracellular fluid that can act as buffers. Proteins, hemoglobin, phosphates, those are found in cells because we also want buffers inside cells. We don't want big changes, pH changes there either. Phosphates and ammonia in urine. We also want buffers in our urine. We don't want our urine to be acidic either. Okay, so how do we control it? So what if the buffers are overwhelmed? Then we have ventilation. So ventilation, or really respiration, gas exchange. So where we can blow off carbon dioxide, and if we do that, then our pH will go up. If we hold our breath and carbon dioxide levels build up, our blood will become more acidic. So we can kind of compensate, or we can kind of change our respiration or ventilation rate to also change our pH. And then we have renal controls. Well, the kidneys, they can just dump hydrogen. They just dump it out in the urine. That's pretty direct and handy, and it reabsorbs more of that, more of the bicarbonate. Okay, so this is a picture. So we're still talking about hydrogen. So the pH scale, this is our pH scale. So if this is our pH over here, and we say that normal pH range 7.35 to 7.45, so that's kind of in this area here. Okay, and then we can say that kind of in the middle of that, 4.8 times 10 to the negative 8 is the pH. It's going to be 7.4, which is just right in the middle. Okay. We have, so if we, I guess if we could look at this really quickly, if we have a pH of 7, so we already kind of looked at it before, but that's 1, 2, 3, 4, 5, 6, 7, 0.0000001. I hope I said that right. And if we increase the concentration a little more, we make it more acidic, so closer to a pH of 6. If we increase, or if we decrease the concentration, less hydrogen, we make the pH go up a little bit, which is what we have. We have it kind of between the 7 and the 8, so it's kind of 4 times 10 to the minus 8, and that's just what it works out to if we do the math. All right, so if we look over here and we look at the concentrations, we have our pH here compatible with human life, 7 to 7.7, but we know that things like saliva is a little more acidic. So saliva here. Urine varies, but it doesn't really get much below 4.5, 4.4. And then we can see other food items. Stomach acid is clear down here at 1, so that one's kind of relevant. So that's mostly just hydrochloric acid, which is very, very acidic. Okay, that hydrochloric acid, it's just like it's handing that hydrogen out to anybody who wants it. As soon as it gets into solution, that hydrogen is free. Okay, so that's why when you put this into a solution, it really causes it to be acidic. All right, so blood pH. So terminology. This is kind of a nice little graph if you like it. It kind of looks at the hydrogen ion concentration increases, and then how that kind of reflects back here on the pH. But acidemia, so really emia means in the blood. So acidemia is acidic blood or blood plasma. And let's see, alkalemia is alkalotic or alkaline blood plasma. It's 7.45. So that's what our range is. So acidemia is less than 7.35. Alkalemia is greater than 7.45. So we can kind of see that reflected in this graph. Okay, so really quickly through blood buffering. So a buffer is defined as a mixture of a weak acid and its conjugate base. So a weak acid is going to have a hydrogen that can be given up, and its conjugate base, so that means the base is going to have a kind of weak acid here where if a hydrogen needs to be donated, it can be donated. Okay, so this will take on a hydrogen if need be, and this part of it will donate a hydrogen if need be. Okay, so we don't need to get into the details of the chemistry. We just need to know a few things, like first of all, we have buffers, and those buffers will operate in a range that's kind of close, a little on the acid side of what our pH of blood is. Like our blood buffers will kind of help us maintain that blood pH in that normal 735 to 745 range. So a buffer makes a solution resistant to pH changes. It doesn't make it so you can't change the pH. It just makes it resistant. If you give acid, then it's going to be like, okay, I have bicarbonate in the blood, so yeah, I'll take your acid and I'll neutralize it. Okay. Or if you have a very basic solution, your blood buffer will be like, okay, I've got some extra hydrogens here. Sure, I'll donate those to you, and we'll kind of neutralize that. So that's kind of what it's doing. And usually the molecule or the chemical that's used as the buffer, in our case, like carbonic acid and bicarbonate, usually those kind of work in a certain range of pH. Okay, so, and we're going to see what that is. So buffering is it occurs in that particular pH range. It's determined by something called the Henderson-Hasselbalch equation. So we can kind of have PTSD from chemistry when we learn this. And it's really just kind of trying to figure out the pH at which it either takes on a hydrogen or it loses a hydrogen. Okay, so where is it? Where is that kind of, I don't know if it's an inflection point, but it's an equilibrium point where the hydrogen and the base are kind of at an equilibrium. Okay, so we're going to see a graph on the next page that kind of explains this a little better, but it's saying, okay, at what pH am I going to take on a hydrogen or give up a hydrogen? Okay, and what range is that going to be? What pH is in the middle, and what's my range? So essentially the equation predicts the pH at which hydrogen is removed from the molecule, so that's a dissociation constant, or the pKa, for A for acid. For example, hydrochloric acid is a strong acid with a low pKa because it gives up its hydrogen very easily at a low pH. So if we look at our blood buffer, so we have a bicarbonate buffer and a phosphate buffer. Okay, so those are the main chemical buffers in the blood, okay, or in the extracellular fluid. So bicarbonate, we have that over here. It's the green one, and we can say that that equilibrium is right here. And on either side of that, we have a kind of a resistance to change. Okay, so so there's not much change in this area. So we can kind of increase. We can add from this point really to this point. We can add quite a bit or take away quite a bit of acid or hydrogen, and the pH will only change a little bit, kind of this range here. Okay, and we can kind of see the same thing for the phosphate buffering. So, you know, kind of in here to here, here. So we can see that range from there to there. We have quite a bit of, on our y-axis, the addition of hydrogen, but really a change is only just kind of minimal within that range. All right, now if we look at this, we can see that this is not our normal pH. Our normal pH is kind of over here in this area, okay? Oops. And really that has to do with the bicarbonate and carbon dioxide levels that we maintain in the blood. Okay, so, okay. And there are other buffers that are also kind of present that help this out. Okay? Because one thing we know for sure is that these are the pKa's, so these are the ranges that these two primary buffers, the major buffer of course being the CO2 and bicarbonate, these are the ranges, but we also know that our pH is over here. Okay, so 7.35 to 7.45. All right. So, so some reasons why are, first of all, yeah, there are other buffers that are kind of present. Proteins can kind of help buffer things. And carbon dioxide is kind of low solubility, so it doesn't pull as much as it could, I suppose. So the low solubility of carbon dioxide kind of maintains the pH at 7.4. But also our kidneys are constantly working to kind of get rid of acid and kind of nudge it over into this range. Okay. Something about the phosphate buffer. The phosphate buffer kind of has a lower impact on extracellular pH. Not because, I mean, it has a lower impact, but because concentration is lower, but also because carbon dioxide and bicarbonate levels vary so much and are so well controlled. Carbon dioxide with ventilation, bicarbonate is more renal. So there's kind of a push and pull of bicarbonate and carbon dioxide that kind of helps keep us in that more neutral range. Okay, and so we kind of see that's what this graph is kind of talking about. So if we look at so if we remember 35, and I keep writing it, hoping that we'll just kind of naturally remember it. But normal carbon dioxide levels are 35 to 45. Okay, so 20, 30, 35 to 45. So this is our range of normal, and I think it's supposed to line up with that, so we'll just make it cheat a little bit. Okay, and then so that's our normal range of carbon dioxide, and then 22 to 26 is our bicarbonate, HCO3 minus. That's our normal bicarbonate, so I'm just not even going to be subtle about it. I'm going to say that's about from here to about here. Okay, so that's our sweet spot. So our sweet spot is kind of in this area right here. Okay, so what does this mean? Well, if we have the ratio of CO2 and bicarbonate, HCO3, if we keep that ratio at the concentrations that are normal in the blood, then we end up with a target right here of 7.40. Okay, 7.40. So anywhere along this. Now, what does that mean, anywhere along this? That means that if you hold your breath, okay, you hold your breath and you are building up carbon dioxide, and your carbon dioxide levels, let's say they build up to like, I don't know, we can just say 50. That means that we're all the way over here. Now, depending on what our bicarbonate levels are, let's say our bicarbonate levels are normal, okay, then you have a pH of 7.30. You are acidotic. That's acidosis. So the only way to fix that, or one way to fix that, so let's say that this is just the way we breathe, this is the way we're going to do it. The only way to get that in a normal range is for your bicarbonate, got to see my numbers down here, to kind of increase all the way, oh gosh, I don't know what that is, 24, 26, 28, 30, 30. Let's see, so it's 6 more, 6 less, so this is going to be 30. Okay, so we'll just say that it's 30. So that means your bicarbonate level is going to have to be 30 to maintain a normal pH. What is the theme of this? Well, this is what we're going to end the whole chapter with. That if someone has respiratory problems where they can't breathe, they have COPD or emphysema, bronchitis, something, and they can't get enough, they can't blow off enough carbon dioxide, their carbon dioxide levels start to increase, well then their renal system is going to have to increase the bicarbonate to help them maintain a normal pH. Okay, so this ratio, now we're not going to be able to live, you know, if carbon dioxide levels are clear up here, so some of these are kind of silly, but it kind of gets that point across that when our carbon dioxide levels go up, well then our body better do something to keep our pH in that normal range. So if our carbon dioxide levels go up, then we're going to have to increase the bicarbonate levels to keep us in that, to keep us at a normal pH. Okay? All right, so note that bicarb and CO2, they kind of work together to alter that pH. All right, so other buffers, organic phosphates, okay, so think adenosine triphosphate, ATP, ADP, GTP, guanosine triphosphate. So those also can function as buffers. Proteins inside cells, particularly those with a histidine residue, or so I've seen. So overall, the amino acid residues, or those little side parts, have a large number of acidic or basic groups. So it depends on which amino acids are present. Some are more on the acidic side, some are more on the basic side. I don't remember them all now. but it kind of depends. But for the most part, most proteins will kind of accept hydrogens. Okay, so they're more hydrogen acceptors than donors. Okay, so that's intracellular. So it's where we see the organic phosphates and proteins. I will say that proteins can also be outside, and in fact, I will say it right here. Okay, because it's written. So proteins can also act as buffers in blood plasma. So for example, albumin can bind either hydrogen or calcium. So if we have our albumin, and it can bind calcium, or it can bind hydrogen. Okay, and so it's a protein, it's not a cloud. And so if hydrogen levels are high, then it will bounce calcium off and the hydrogen will take the place of the calcium. If calcium levels are high, then it'll bounce that hydrogen off and it'll take the place of the hydrogen. So basically what that means is they're both kind of competing for the same binding sites. And if you're, so, okay, so in acidemia, so when you have high hydrogen levels, okay, we're acidic, so our hydrogen concentrations are high, then what you're going to do is you're going to be bouncing the calcium off. So that's mainly what we think of or talk about is the effect that hydrogen has on calcium levels. So during acidemia or acidosis, then you may also see hypercalcemia or high blood calcium levels. Okay, so this is kind of a reminder with a specific example that that everything's connected. So our acidic levels will cause calcium levels to go up, and in the same way, high hydrogen levels will cause potassium levels to go up. Okay, because as we'll see, there are hydrogen potassium pumps at work in the body. Chloride is the same way. So chloride but it competes with bicarbonate. So we have these connections between some of these ions. In this case, we're talking about the effects on proteins with calcium and hydrogen. So this is protein related. My other examples are, we'll kind of see where those come into play, but those are kind of inside and outside of cells. Okay, so plasma proteins can act as blood buffers, and I kind of got off on a little tangent there that I'll return to later. Okay, so renal mechanisms in acid-base balance. So the kidneys have two major roles in the maintenance of acid-base balance: reabsorption of filtered bicarbonate. So we've been talking about it, that the kidneys reabsorb as much bicarbonate as they can until it gets to about like 40. Remember, the range is normally like 22 to 26. So it kind of will keep reabsorbing back into the body bicarbonate so that it's not excreted in the urine. So nearly 100% of bicarbonate is reabsorbed until it gets so saturated that it can't be. And not only that, but we actually form new bicarbonate. We produce bicarbonate in our body through that, that reaction to make the H2CO3 to make HCO3 minus. So our reaction, our blood buffer reaction with bicarbonic acid, will make more of it and then just store it in the body. Okay? Move it back to the body. And then just dump that hydrogen out. So we're going to see how that takes place too. So excretion of hydrogen. So we can't just dump hydrogen into our urine and just walk away. So because that's going to cause our urine to become very acidic, and that's not going to be pleasant. Okay, so we can't just have, in the kidneys just saying just dump the hydrogen. That's kind of, I don't know, we don't expect industrial sites to dump their waste and just say, deal with it. So because hydrogen would cause the urine to become very, very acidic, and then we would be peeing acid. So what happens is it gets bound to things. We have buffers in the urine, so excretion of hydrogen as ammonium ion. Okay, so we stick it to an NH3, and then which acts as a base, and then it accepts that hydrogen, and it can be excreted that way. It's kind of harmless. And then excretion of hydrogen is what we call a titratable acid. Okay, so it's bound to a phosphate group. And that also, it's titratable because I guess you can titrate it and find the acid, but find the hydrogen. But that's another way that hydrogen can be excreted. So both these methods are tied to reabsorption and production of new bicarbonate. So we're going to kind of see that and how that works. So excretion of hydrogen as ammonium ion, the cation, and the titratable acid. So we're going to have a couple of pictures of this. This one's kind of an oversimplification because it doesn't really talk about some of the exchange that happens, but it kind of gives the overall impression of what you're doing. So fixed hydrogen from protein phospholipid metabolism, so it has to be excreted, bound to a phosphate or that ammonium cation, that ammonium ion. So 15%. So remember, phosphates are part of our blood buffer. So about 15% are filtered and move into the renal filtrate. So remember the kidneys and the tubules. So about 15% are filtered through the glomerulus and move into the renal tubules. Now, what happens, so we have production of hydrogen here, and so the hydrogen then can be excreted or secreted out into the tubule where it will bind with HPO4. Okay, so this is hydrogen phosphate, and then to form dihydrogen phosphate. Okay, so it just sticks a hydrogen to that, and then that's excreted in urine. Now, as far as amino acids, breakdown products of proteins, there are lots of different breakdown products of proteins. We also heard about urea and well, creatinine is just another breakdown product, but urea is another breakdown product. So here we have, and kind of in this line, but this is ammonia specifically, and so we can stick amino acids. So we can stick the hydrogen onto an NH3, produce NH4, and then NH4 can simply be excreted in the urine as NH4. Now, I mentioned earlier, and then bicarb, note that bicarb is reabsorbed into the body. Okay, so it's back into the body. Now, I mentioned earlier that the pH of urine can only get as low as 4.4, and that's because the pumping process can't function at a pH lower than 4.4. Okay, so that may happen if there's a low concentration of urine phosphate buffers to absorb the hydrogen. So there are situations where the pH will be very, very low, very, very acidic, and it just has to kind of maintain at that pH until and kind of whittle away at it, because we really don't want that pH to drop much below 4.4, and it really can't, because the pumping mechanisms like the sodium potassium or sodium hydrogen pump here won't function, or exchanger. Okay, so let's look at what we have here. So this is this. Remember, I said that sometimes we will actually make, we'll mix the carbon dioxide and the water just to make more bicarbonate. So this is our goal here, is to make bicarbonate and then to reabsorb the bicarbonate. Okay, so this is our goal. So we use this equation that we know and love, the carbon dioxide and water to carbonic acid to bicarbonate and hydrogen. And then we push the hydrogen out through exchange with sodium. Remember, sodium wants to get in the cell, so we just kind of flip it and we say, okay, sodium's out, and then, or the hydrogen is out, and then it will go away on one of the urine buffers. And then over here we conveniently nicely have the bicarbonate that is exchanged with chloride. Okay, so this is kind of what I was mentioning earlier, where chloride changes can affect our bicarbonate, because we have to maintain that chloride in a certain level so it works properly to help move that bicarbonate out. And then the secondary active for sodium kind of, let's see, oh, the bicarbonate goes out and the sodium goes with it. Okay, so that's not secondary active, that's just bicarbonate kind of moving out and moving the sodium with it. Okay, so let's see. So I think we just went through all of this. So bicarbonate is moved back to the body by chloride exchange and by sodium cotransport. So really, when we look at that, there's no net excretion of hydrogen. So there's no pH change in the filtrate. Okay. So the hydrogen is moving out, but then let's see. But then the equation, I guess here, I said earlier that it might bind to something, but it looks like the equation here is just working in reverse. Okay, so then the carbon dioxide, yeah, if we read through this, the carbon dioxide is pulled back in, reacts with the water, carbonic acid, and then bicarbonate. Hydrogen moves out. Hydrogen will mix with bicarbonate that was in the filtrate. Sorry, I didn't go through this well enough first time. So bicarbonate is in the filtrate, HCO3 minus, mixes with that hydrogen carbonic acid, and then splits to water, CO2. So this just kind of is a cycle that keeps going around. Okay, so that hydrogen is actually kind of using bicarbonate that was in the filtrate, and ultimately that bicarbonate is kind of used up in this equation. But the whole point is to make more bicarbonate to pull back into the body, so we don't lose what's in the filtrate. Okay. Let's see. So changes in bicarbonate, reabsorption. So this is kind of the same thing. So we'll just, but I've added some more information here. So normally bicarb is nearly 100% reabsorbed. At blood bicarbonate concentrations, I think I mentioned this earlier. Remember that normal bicarb is 22 to 26. At blood bicarbonate concentrations greater than 40, the transporters are saturated, so bicarb is excreted. So these processes of pulling the bicarb back in change, and then we have so then bicarbonate will actually be excreted. So this process stops, and any kind of bicarbonate that was in the filtrate will go through and be lost as urine. Okay. So in respiratory acidosis, carbon dioxide is increased. Okay, so that means that hydrogen and bicarb are produced more rapidly. Okay. So what does that mean? So if we look at our equation, then if we have increased carbon dioxide, we have plenty of water, so we kind of push our equation this direction, and we have more production of hydrogen and bicarbonate. Okay, so during acidosis, and so if we increase this rate of the reaction, then we are going to be moving more bicarbonate in. Okay, so this kind of gives us some information. Earlier when we said during respiratory acidosis, the liver or the kidney is going to have to reabsorb more bicarbonate. Well, this is how it does it, because if we have, during respiratory acidosis, we have more carbon dioxide. So that means this whole process, this whole equation is going to happen more rapidly and more freely. Okay, so we're going to have more carbon dioxide available for this reaction to take place. Okay, which then binds with hydrogen in the body. So more bicarbonate binds with hydrogen in the body to get rid of those free hydrogens, which is going to increase pH. Okay, so in respiratory alkalosis, less carbon dioxide is available, so less HCO3 is reabsorbed. Okay, so this process will kind of slow down if we don't have enough carbon dioxide. Okay, so it's kind of a natural little process, slows down, and so that is going to, during alkalosis, will cause the pH to rise because there's less bicarbonate to neutralize those hydrogens, acids, those hydrogen things. Okay, let's see. So this is kind of talking about where things happen differently, but it also kind of mentions something else that's kind of important, and this is the hydrogen potassium pump. So excretion of hydrogen as the ammonium cation, so the NH4 plus. So hydrogen is excreted as the ammonium cation in the proximal tubule. Okay, so we've talked about how this more or less takes place. In the proximal tubule, the thick ascending limb of the loop of Henle, and in the collecting ducts, okay, in those certain types of cells, the alpha intercalated cells. So we have to have an NH3, so we didn't really discuss that earlier. We kind of have to have an NH3 on which to put the hydrogens. Okay, and then NH3 really comes from the breakdown of amino acids, in particular, glutamine. So NH3 is produced by the deamination of the amino acid glutamine, and that diffuses into the nephron. So glutamine diffuses the NH3. NH3 goes wherever it wants to go, honestly. NH3 is not charged, and it's small, so it'll just diffuse through the cell if it wants to diffuse into the cell. Okay, it'll diffuse out of the cell if it wants to diffuse out of the cell. And so it will diffuse into the nephron tubule. So that's kind of what we're seeing here. If this is the, we see the lumen, so this is the nephron tubule over here. Okay, so we've got, our filtrate is in here. And we're saying, all right, how are we going to get rid of this hydrogen as ammonium ion? So we're saying that first things first, we need to get that NH3. We stick the hydrogen to the NH3, and then the the NH3 will move naturally into the lumen. So if we're wondering how the NH3 got there, then it can just freely move there because it is not charged. Okay. Hopefully that makes some sense. Okay. So hydrogen then is excreted by the sodium hydrogen exchanger. So now we have hydrogen out here, which will stick to that NH3 to produce the NH4, hydronium ion. All right. Okay. So glutamine produces the NH4, removes the hydrogen. The hydrogen then has to move through separately. NH3 diffuses through. So that's in the proximal tubule, and we're not gonna bother ourselves too much by noting these differences, but more the process of how this takes place. So okay, so in the collecting duct, again, hydrogen is exchanged with potassium by the hydrogen potassium, or not again, but we have hydrogen. Let's see here. We have kind of a new player here, the hydrogen and potassium exchanger. So potassium moves into the cell while hydrogen is pushed out of the cell. This is an ATP-driven process. Okay. All right. So what we've done is that we have a buildup of hydrogen. Okay. And so we have to get it out, and we're trying to put it into the lumen. And again, whereas our NH3 in the proximal tubule was kind of produced in the cell and then diffused out, in this case, so it comes from the interstitial fluid from the kidney, and it just diffuses right into the lumen as well. So either way, NH3 is diffusing into the lumen to act as a proton acceptor or hydrogen acceptor. Okay, and that's really kind of the whole point of both of these. It's just saying it's done in a little bit different way in the distal tubule as in the proximal tubule. And I don't know, I like to point out things like the hydrogen potassium exchanger and bicarbonate and chloride, because you'll see that potassium has an effect. So high potassium levels will have an effect on hydrogen. So when potassium levels are higher, then your hydrogen levels will also be higher, because what happens is that potassium will move into the cell and it'll kind of displace that hydrogen and push the hydrogen out. Same thing with chloride. Chloride depletion will kind of prevent that bicarbonate from being exchanged too. So you can have chloride depletion, pH changes, potassium acidemia or acidosis caused by hyperkalemia. So just, when you start talking about that stuff later and these pH changes, just kind of remember that a lot of this comes from these exchange mechanisms that are happening in the body in general, but especially here when you're trying to change the pHs, okay, or modulate the pH in the kidneys. Okay, so let's see. Acid-base disorders. Disturbances in blood pH can be caused by a disturbance of bicarbonate or carbon dioxide. So we're going to kind of get into our ABGs, our arterial blood gases, and how to kind of, predict whether we have an acidosis or an alkalosis, or whether it's respiratory or metabolic. So we'll start talking about that stuff. So disturbances in the blood pH can be caused by a disturbance of these two, the bicarbonate and the carbon dioxide. So we've kind of discussed that, and we've seen our equation here. So disturbances lead to acidosis or alkalosis, or what the books, or what we've been calling also acidemia or alkalemia, so acid blood or alkaline blood. And then, yes, we classify them as either metabolic or respiratory, depending on the source of the acidosis or alkalosis. Okay, so are they respiratory in nature? If they're respiratory in nature, that's because if it's acidic, then it's because your carbon dioxide levels are building up because you can't blow off your carbon dioxide. If they are metabolic in nature, that just means that they're not respiratory. Okay, so metabolic can come from a whole lot of sources. It could come from, kidney injury, or it could come from, by kidney injury I mean like kidney failure, or it could come from just vomiting, diarrhea, lactic acidosis, anything like that. And I'll show a couple of pages of some examples as we go along. All right, so compensation. So if we have a respiratory problem, well, okay, so let me just say this first of all. First of all, our compensation for high acid or low, or high hydrogen or low hydrogen, or acid or alkaline influences are the buffers. So the buffers are the first line of defense. So you've got these buffers that are kind of just in your blood, and they're constantly doing their thing. And minus plus H plus. So that's always in the blood, and so they're or extracellular fluid, so they're always kind of working. And then we have respiratory compensation. Respiratory compensation is if your blood is too alkaline, you hold your breath. Okay. So if you have alkalosis, you don't hold your breath, like really, you don't have to think about it, but your body will naturally sort of slow the ventilation rate to kind of compensate for that. And the same in the other direction. If it's very alkaline, well, let's see, if it's alkaline, you'll hold your breath to make it more acidic. If it's acidic, then your ventilation rate will increase. So somebody with like DKA, their ventilation rate is very fast. Okay. So then we have renal compensation. Well, if respiratory system is the cause of it, then the kidneys are responsible for either dumping more hydrogen or not dumping hydrogen, really. Okay, so if the acid base, so we can say this next part, we can say this in general, and this is accepted and it's true. So if the acid base disturbance is metabolic, then your compensation will be respiratory. Okay, so if something is happening in the body that is changing the pH, then that will change our ventilation rate. It'll change the respiration, the respiratory response. Okay. Now it may also, if it's not renal in nature, the kidneys may also get involved, but it will definitely be a respiratory response if it's a metabolic problem. Okay. So if the acid-base disturbance is respiratory, then the compensation will be renal, and really that's kind of I mean, if it's respiratory, we already know the source of it. The source is the lungs, and so we kind of have to have. We can't have a respiratory response. It's going to have to be a renal response to that. Okay. So I kind of said the same thing here. I did point out that could involve, so compensation for metabolic acidosis could involve renal mechanisms if the kidneys aren't the cause of the problem, like if it's vomiting or diarrhea, something like that. Then yeah, the kidneys can kind of get involved with that as long as they're functioning. But you'll definitely have a respiratory response. All right, so let's see. Summary of acid-base disorders, metabolic acidosis, metabolic alkalosis. This is going to see a change in bicarbonate, so we have the thicker lines here. You'll also see opposite, or the directional changes of carbon dioxide levels changing, but really when we're talking about metabolic, we're talking about bicarb, and when we're talking about respiratory, we're talking about carbon dioxide. Okay. And, we'll see, when during respiratory acidosis, when carbon dioxide mixes with water, ultimately it will produce more bicarb. It will have, if it's alkalosis and it slows down, it'll be less bicarb. But that's minimal compared to the response or the effect that it has on, in the case of respiratory, the effect that it has on carbon dioxide. And if it's metabolic, the effect that it has on bicarbonate. Okay, so compensation, so check these out. Hyper or hypoventilation, depends on which direction the pH is. Hopefully we're reading that. And then the renal compensation will be to either increase bicarbonate excretion, reabsorption. Yeah, okay. Okay, so So next, okay, so one other thing to talk about before we, I guess, move on is part of the diagnosis of what's going on, whether, for metabolic, especially, well, pretty much only metabolic acidosis. So there's this thing called, this lab test, it's called anion gap. Anion gap, which is written here as well and here. And the premise of this is that when you do a lab test, there are certain things that are always measured, okay? And there are some things that aren't measured most of the time, okay? Things that are always measured tend to be sodium and chloride, and I guess we could say bicarbonate. So those things are measured, and we can see those. And when you look at, and note that sodium is positively charged and bicarb and chloride are negatively charged. We also look at potassium, but potassium is going to be very small amount. So like if we were to add potassium to this, it might be just barely add anything at all. So a lot of times we just ignore the potassium. And we think, okay, now in our blood plasma, that's what we're talking about, it's neutrally charged. Okay, so I know that we've talked about how inside versus outside of cells, the inside of the cell is negative compared to the outside of the cell. But in our blood plasma, it's a neutral solution. Okay, so there's no net charge of the solution itself. Okay, so that means the positive charges equal the negative charges. So if we look and we measure sodium, and then we measure chloride and bicarbonate, there's a part up here that we don't measure, and that's a gap. Okay? So there's this space up here that we say, okay, sodium doesn't, the positive sodium, okay, remember we're ignoring the potassium, doesn't match the negative, the chloride and the bicarbonate, and it never does. Okay? So there's always a gap, and in that gap are maybe some organic anions. Okay? So for example, it could be, lactic acid, when it gives up its hydrogen, gets to a lactate anion, and that's negatively charged. Okay, ketones. Ketones, when they give up their hydrogen, they are negatively charged. So if we see an anion gap, we think, okay, that's normal. So there's a normal range for an anion gap, and I meant to look that range up. I can't remember what it is. And so there's a normal range, but when we see that range kind of getting bigger, then we say, okay, then these things that we're not measuring may be building up. Ketones may be building up, lactates, anions may be building up. And so that's kind of an indication that, especially if the blood pH is off, that we have a metabolic situation that is that's causing a buildup of these anions, maybe these organic anions. Okay, so that's going to increase our anion gap. So if someone is in shock and their lactate is at high concentrations, negatively charged, their pH is decreasing, so their pH is low, and they have a large anion gap, then that may indicate lactic acidosis. Or in the case of like diabetes, I mentioned earlier that a lot of times glucose can't get into cells because insulin is not present, and so the inside of those cells are producing ketones, and those ketones are acidic, and when they lose their hydrogen, they're negatively charged, and so those may be building up. So it could be diabetic ketoacidosis. Okay, so the ketones could be building up. Either way, you've got an increase in that gap, that anion gap. All right, so let's kind of read through this. So measured ions include sodium, bicarbonate, chloride, also potassium, but it's really low. Measured positive charges will increase to keep the plasma neutral. So we're trying to keep that plasma neutral. Sodium will always have a higher value than just bicarb and chloride because there's always something. There are always unmeasured anions that make up the difference. So let's see some examples listed here. Proteins, phosphates, citrate, sulfate are some of the mystery anions. So if the unmeasured anions are increased, then sodium will also be increased to offset measured organic anions, and the anion gap will be higher. So we're always going to kind of keep this at the same level. It's just what proportion of this is the gap. Okay, and so if our anion gap is increased. Okay, let's go to the next slide. Same thing. So metabolic acidosis with an increased anion gap may be what we're looking for. So a decrease in bicarbonate will result in an increased anion gap. Low bicarbonate usually indicates a metabolic acidosis. Okay, so in metabolic acidosis, there may be an accumulation of the organic anion, which is going to cause metabolic acidosis with an increased anion gap. Okay, so organic acids can donate the hydrogen to the bicarb, leaving a negatively charged organic anion and reducing the bicarb in the blood and increasing that gap. Okay. So the organic acids that we should really be thinking about really come down to the negatively charged ketones, so diabetic ketoacidosis. So this is a good indicator of diabetic ketoacidosis. And then we can just look for hydroxybutyrate, I think, which is a type of ketone that we can measure and see if that's building up. Beta hydroxybutyrate, I think, is one of the ketones that we can look at. And if that's built up, then we can say, okay, well, that explains the increased anion gap. Lactic acidosis, so the negative lactate anion, which is associated with shock or something else that may be causing the hypoxia, some kind of a hypoxic situation, lack of oxygen. Shock, it's usually just low blood pressure, and our organs are starving because they're not getting enough blood perfusion. So, and then there's some others listed here: formic acid, methanol poisoning, oxalic acid from ethylene glycol poisoning. Okay, but really a lot of times what we're looking at is really diabetic ketoacidosis. Okay, can be an indicator of increased anion gap or lactic acidosis as well. Okay, so let's see. Acid-base imbalance, respiratory. So respiratory is the source of pH changes. That's respiratory acidosis. Respiratory alkalosis is, so acidosis is acidic, respiratory alkalosis is alkaline. We've mentioned this before, so I'm going to kind of cruise through it. So metabolic is when the pH change is caused by something other than respiration, so kidney failure, shock, diarrhea, vomiting, etc. So we have metabolic acidosis, metabolic alkalosis. And yeah, we just saw that we can also have like DKA, diabetic ketoacidosis. That's a type of metabolic acidosis. So determining this requires these values. So maybe we've memorized these by now, maybe we haven't, but pH 7.35 to 7.45, carbon dioxide levels 35 to 45, partial pressures, and then bicarbonate 22 to 26, and that's in milliequivalents per liter. Okay, those are the units. But these are the numbers, okay, and the numbers are very important. So let's just kind of look at a few things, kind of cruise through these, and then we'll do some examples. So acidosis, pH is less than 7.35, alkalosis greater than 7.45. So acidosis can come from increased acid or decreased base. So respiratory, so some of the things that may cause acidosis, hypoventilation and retention of carbon dioxide. It could be from sedation. So it could be an accidental sedation. You took too many opioids. Central nervous system disorders that prevent proper ventilation, neuromuscular diseases, low oxygen environment, or any number of lung diseases or infections can cause acidosis as well. And these are respiratory. So these are all lung related. Metabolic, so this can be a loss of bicarbonate. So that can come from severe diarrhea. So increased acid production, and these are these organic acids like the ketone acids, beta hydroxybutyrate. I hope I'm saying that right, because I'm remembering it from a time. But you can look it up, you'll remember it too. So diabetic ketoacidosis, shock, anaerobic metabolism, which is lactic acid. Both shock and anaerobic metabolism will cause an increase in lactate anion, which is a metabolic acidosis. Alkalosis, increased base or decreased acid. Respiratory, decreased acid secondary to hyperventilation. So you're blowing off carbon dioxide, you're breathing too fast. Fear and anxiety can cause that to happen. I can tell you that I can tell you a weird little story. I was scuba diving, and I had only scuba dived a couple of times, and we just rented a boat, and I trusted it, I thought. But anyway, as soon as I got into the water, you usually have a spare respirator, the thing that goes on in your mouth so you can breathe. Well, anyway, mine popped off as soon as I went into the water, and I took in a bunch of air. Well, the people running it just told me to use my spare. So the whole time I'm down 30 feet, I'm not an old hand at this, and I don't have a spare. So I was like breathing very, very, very quickly. I had a lot of anxiety. I used up my oxygen very fast. I didn't have I don't know why I'm telling the story now that I think about it, but I didn't measure my blood pH, but I was definitely blowing off a lot of carbon dioxide. And so that was my little example. But I don't know why I told that. Just wasted a whole lot of time. But anyway, most people, it's going to be like panic attacks, fear, anxiety types of responses. For me, it was thinking I was going to die in 40 feet of water. All right, metabolic, increased reabsorption, so that was respiratory. Alkalosis. Metabolic, increased reabsorption of carbon dioxide, so ingestion of large amounts of bicarbonate and acids, which happens. A lot of people will continue to take those, and that can really alter your blood pH. Excretion of hydrogen, so severe vomiting, acidic urine production, that kind of thing. All right, so more examples. We'll just kind of read through these because some of them are repetitive. Increased levels of ketoacidosis from lipid breakdown, lactic acid from anaerobic cellular respiration, metabolic production of sulfuric acid, phosphoric acid, and decreased bicarbonate. Alkalosis, yeah, so these are just kind of saying the same things. I guess one added here is kidney and GI malfunction. Okay, so but we kind of know that. So if the kidney isn't working right, then that's going to be a metabolic cause of whatever the kidney is not doing right. But when the kidneys fail in general, they're not going to be bringing in, they're not going to be dumping hydrogen as well. Okay, so that hydrogen, or they're not going to be bringing in the, let's see, yeah. So metabolic alkalosis. Either way, somehow they're bringing in too much bicarbonate and not releasing too much acid. So the kidney malfunction is going to cause blood pH changes in either direction. But really, I think of it as you're not going to pull in as much bicarbonate, so it's actually going to be more of an acidosis kind of a thing. So let's see. So what are we looking at? I've simplified this. This is our calculation, our chemical equation, and it shows respiratory acidosis and respiratory alkalosis. So in general, with respiratory alkalosis, what we are looking for is increased carbon dioxide. Alkalosis, we're looking for decreased carbon dioxide. So the take-home message here is respiratory, you're looking at partial pressure of carbon dioxide. That will cause an increase in hydrogen, which is what is causing the acidic condition. Okay, and it's the opposite with alkalosis: decreased hydrogen, free hydrogen, and increased pH. Okay, so we see the same thing with metabolic in terms of the pH, but what we look at with acidosis is the bicarbonates. So acidosis, we're going to see decreased bicarbonate, which is going to, if bicarbonate is decreased, we're going to see an increase in hydrogen, okay? And that's going to cause our low pH. Okay, so that's metabolic acidosis. Metabolic alkalosis, increased bicarbonate, okay? So we see more bicarbonate, and when we see more bicarbonate, it is going to bind with the hydrogen, and the hydrogen is going to decrease, and that's what's going to cause our pH. Okay, so we have a few different things that are going to happen that we've already kind of discussed. So if we have a situation, so initiating event, okay, so the causative event, the initiating event, so whatever caused this. So a couple of examples here is ketoacidosis for metabolic acidosis, okay, change in respiration, whether increase or decrease for respiratory alkalosis or acidosis. So whatever the initiating or causing causative event is, that's our primary mechanism. So the underlying cause of the imbalance. Well, then we have compensation. Okay, so the compensation, if it's respiratory, it's going to be a renal compensation in general. So mechanism is going to be different than that which caused the imbalance. Okay, so we're trying to adjust the pH by, if it's metabolic, if it's caused by kidney damage or kidney failure, then we're going to try to fix it through respiration, or anything metabolic. We're going to try to fix it through ventilation rates. Okay, so that's our compensatory. So it attempts to adjust the pH towards a more normal level, but you're not really correcting the problem. So if the problem is emphysema and you're trying to correct it with the kidneys, you're not fixing the emphysema. Okay, so you're not getting better gas exchange in the lungs. But that's a compensatory mechanism. So it's a temporary effect. You're not fixing the problem. So it can either be compensated, partial compensated, or uncompensated. If it's uncompensated, that just means, and this is the thing to remember here, if it's uncompensated, you haven't compensated. Your pH is still out. Okay, so you're either still below the 7.35 or above the 7.45. You're still out of range. Your causative agent, whatever's causing it, is still not working. So our pH is not working. The compensating system is fine. So let's say that my pH is 7. I can't breathe. Whatever, for whatever reason, my pH is 7.25. That's very acidic. But my my bicarbonate is fine. My kidneys don't even care. Sorry, they do. They just may not be responding yet. But the compensation still remains normal. So 22 to 26. So maybe my bicarbonate is still 24. So that's completely uncompensated. So nothing is happening. My pH is out. I still can't breathe, and my kidneys they don't even care. Kidneys don't love me. So, but they may not have started responding yet. So that's the situation we're in. So the kidneys haven't done anything yet. Okay. All right. Or at least they haven't changed the bicarb levels yet. All right. So partial compensation. Well, we're getting there. Okay. So now what did I say? My pH was 7.25. So I'm acidic. I can't breathe. So it's respiratory. Well, now we're in partial compensation. So my pH is still, maybe now it's like 7.3. I don't know. Or it's still 7.25, whatever. But then you look over at the at my bicarbonate, and my kidneys are coming through for me. So my bicarbonate normal is 22 to 26. My bicarbonate is clear up here at 28. Okay, so it's out of range, which means it's saying, hey, I got you. We're going to fix this. So the bicarbonate levels increasing are trying to increase my pH. Okay, so I'm fixing it. So this is acidic. This is from high carbon dioxide. My carbon dioxide levels are high, but now my bicarb levels are also high. Okay, so I'm getting there. I'm doing it. I've got partial compensation. Okay, so partial compensation. Yeah, my pH is still out of range, so pH is not yet returned within normal range, but I can see from my ABG, my blood gases, that my bicarb is fixing the problem. Okay, so that's partial compensation, and then complete compensation means your pH is within normal range. Okay, so now my pH is back up to 7.36. Okay, my bicarb levels may still be high. My carbon dioxide levels may still be high, but hey, I have compensated. Okay, and so that's good. At least my pH levels are normal. I still need to fix the problem, but my pH levels are normal. All right, so let's look at some examples. I'll go through. I have like 10 of these. I'm not going to go through all of them, but I'll try to get to at least one that's either partial or partial compensated. All right, so pH is 7.3. So if we go through this, pH is 7.3. If our normal is 7.35 to 7.45, we know that that is acidic. So what we do up here is we say, okay, here's the acidic side. That's definitely pH. So the pH is on the acidic side. And then we say, okay, so what's going on here? Okay, well, my carbon dioxide level is high. Okay, so it's supposed to be 35 to 45. This is at 50. Okay, so that's high. So we're going to say that's also on the acidic side. So CO2 is on the acidic side. My bicarbonate, 22 to 26, is on the normal side. So my bicarb, I'm going to put here. So I can already see that I have, some people would call this a tic-tac-toe. We're on acidic, pH, carbon dioxide. So that is going to be respiratory acidosis. Okay, pH is out of range. Okay, so there's no compensation, and it's uncompensated. Now, one other thing we can look at here is we can look at, okay, are we partially compensated? No. Bicarb's not doing it. It's not there yet. Okay, now if this bicarb were like 28 or 30, then we would put it over here in the basic side of things. Okay, so we put our bicarb over here, and we could say, okay, it's pulling, so it's trying to compensate, but it's not there yet. Okay, there's no partial compensation. All right, so let's go through and see if we did it. See if we did it right. Actually, it's not to see if we did it right, to see if I made the charts correct. So got the same thing. So acid, bicarb is normal. So we can say that this is uncompensated respiratory acidosis. Okay, so that's correct. Okay, so now we have a pH of 7.58. So I'm going to just go ahead and put this over here because that is alkaline. So 735 to 745 is normal. This is definitely on the alkaline side. My carbon dioxide is low. Okay, so remember, carbon dioxide, when it's high, that's acidic. When it's low, that's basic. Bicarb, it's the opposite. Okay, it goes the other direction. So when bicarb is high, that's alkaline, and when it's low, it's acidic. So so we can see that the carbon dioxide is going to be over here in the alkaline carbon dioxide, and our bicarbonate 22 to 26, it is normal. Okay, so HCO3 minus is in the normal phase. So we can see that this is alkalosis. We can see that it's because it's carbon dioxide, that it's respiratory. And we can see from our pH that it's uncompensated. Well, we can see from our pH and the fact that our bicarb is normal that it's uncompensated. It's not partially compensated because then our bicarb would be pulling it. So this is uncompensated respiratory alkalosis. Okay. So let's see if I did it right in the thing. Yes, so we're over here, normal bicarb. So uncompensated respiratory alkalosis. So let's look at this one. Oh, look. So we have a pH of 7.38. Okay, so that's normal. So our pH is normal. Carbon dioxide is high, which is on the acidic side. So we're going to say carbon dioxide is over here, and bicarb is low. Okay, well that's weird. Okay, so the bicarb is high, indicating alkaline. So bicarb HCO3 minus. So where's our tic-tac-toe? Well, so in a case like this, we would look at which side. So even though our normal is 735 to 745, we base it on 7.40. So that's perfect. If it's lower than that, we say it's acidosis. Just to kind of give us our indication of where we started from. So we are still on the acidic side, and then alkalosis would be if it's over 7.45. So we're less than, or over 7.4. So we're less than 7.4. So we're going to put our pH over here. Okay, so it's normal, yes, but it's on the acidic side. So we're going to say that, so if we ask the question, first of all, what do we have here? We have respiratory because it's being caused by carbon dioxide, so it's respiratory. And it's acidosis. But because our pH is in the normal range, it's compensated. So good job, kidneys, you have fixed the pH, compensated. So respiratory acidosis compensated. So same thing, same process. And then fully compensated respiratory acidosis. I just didn't move that pH. So you can do that. You can move the pH over here and say this is it's on the acidosis side of things. Okay, a couple of more examples. Let's see. So we have pH 7.39. So that's going to be in the normal range pH. So whatever this is, it's, unless everything else is normal, this is compensated. So carbon dioxide, yeah, no, that's already on the alkaline side of things. So CO2. And the bicarbonate is definitely on the acidic side of things. Okay, so do you kind of see what we're doing here? When carbon dioxide is high, that's acidic. When bicarbonate is high, that's alkaline. When carbon dioxide is low, that's alkaline. And when carbon dioxide is low, it's alkaline, yeah. And when bicarb is low, that is acidic. Okay. So this is acidic. So we're less than 22. Bicarb is low, so it's on the acidic side. If we do our little trick and we say, okay, well, yeah, you're normal, but you're really trying to be acidic, so we put our pH over here, then we can see that we have metabolic, because our change is not respiratory. Okay? Our change, our acidic side of things, is bicarb, and it's acidosis, and it is compensated because we do have a normal pH here. Okay, so pH, let's see, yep, move it over there, and then fully compensated metabolic acidosis. Okay, so let's look at this one. Let's see, I'm not sure where we're going to go with this. 7.39, so I'm just going to go ahead and fill this in a little more quickly. So yes, it's normal, but also it's on the low side of normal, so we're going to put it over here. In the acidic side, our carbon dioxide's 35 to 45, that's going to be high, which is going to cause acidic, okay? And our bicarbonate is low, okay? So that's going to be, let's see, ooh, that can't be right. I think I knew I had at least one of these that was messed up. Okay, let me see if I changed the next one. Okay, yeah, I did. Okay, so I screwed up here, and this should be a 3. So bicarb is 30. So we are on the basic side of things. So hopefully we don't. Now that's one thing we aren't addressing is, are there two causes? And so that requires something, I think it's called like the Wolff equation or something like that, that you have to expect. You have to kind of indicate what the expected compensation is. And then if you're kind of falling into that range, then you can say, okay, that's all it is. But sometimes you have, you may have a respiratory problem and you may have a metabolic problem. We're not addressing that right now. That'll happen hopefully maybe sometime later. Okay, if you go into like pulmonary or renal, I don't know. So anyway, so our bicarb is actually over here, HCO3 minus. So our bicarb is over here. And so we can see that we have respiratory acidosis, and it's compensated. Okay, so again, there we go. We see our pH is actually over here, and we actually see that our bicarb is supposed to be 30, and fully compensated respiratory acidosis. So we have to do at least one more because we want to see some other stuff. All right, so pH 7.32. Okay, so we're going to move quickly here. We're going to say this is acidic because it's less than 7.35. We look at our CO2 levels. It's higher than 4.5, so we're going to put it also over here, CO2. And we look at our bicarb, and we can see our bicarb is definitely over here on the basic side of things. Okay, HCO3 minus. Okay, so this is a different situation. Okay, so what we see here is that our initial problem is not corrected, and our compensation is also out of range. Okay, so we have, so what that tells us, and this is kind of an important lesson here, is that our kidneys are trying. Okay, so this is definitely, so we've got our tic-tac-toe, whatever, over here. So we definitely know that this is respiratory acidosis. But our kidneys are working on it. So it is partially compensated. Okay, why isn't it fully compensated? Because our pH is still out. Okay, so it's partially compensated. So partial compensation. So that should be what we find. Yeah, we don't have anything normal. And so we have our bicarb over there, kidneys working hard, saying, hey, I'm giving you the bicarb, I'm doing my best. So that's partially compensated respiratory acidosis. We give, some nice kudos to the kidneys for that. All right, one more, and then we'll stop. All right, so pH 7.25, acid. Actually, you know what? Yeah, we'll do this one. We'll do this one and then quit. All right, 7.25, so that's going to be acid pH. This can be fun after a while. Carbon dioxide is 55, so that's also CO2 is over here. Bicarbonate is normal, so bicarb is in the normal. Wait, no, it's not. Yes, it is. Yes. So bicarb is 25, so yeah, that's right there in the normal range. Okay. So that is, I mean, this is kind of boring. So we've got, it's definitely, you know, respiratory. A lot of respiratory acidosis is here, and it's uncompensated. Okay. So uncompensated respiratory acidosis. Okay. All right. Well, I'm gonna stop with this stuff, and I'll leave you a few examples to just kind of see it do on your own. Okay. Have a good day.